Hello, I'm Lisa Tucker-Kellogg from the Duke-NUS Medical School in Singapore. Today I'll talk to you about computational modeling to predict the evolution of drug resistance in populations of cancer cells. We're looking at how evolution of drug resistance is affected by the amount of synergy or additivity in a combination of drugs. The goal is to predict the future, which treatments can best lengthen the time span of disease control and evade drug resistance. To do this, we make some simplifying assumptions. Although I do love network models, today we're skipping molecular mechanisms and looking at the simplest possible models, just the number of cells over time. If we have some drug-sensitive cells in blue and a drug-resistant cell in red, then our minds can imagine what will happen during drug treatment. That means our mental models can predict the future, at least qualitatively. The exact number of cells would depend on the details of the system, and much of this variation can be summarized using just a few empirical readouts: the proliferation rate and death rate for blue cells and red cells, and a mutation rate for cells to change state. I hope you're willing to believe that this is enough information to write a mathematical equation or a computer program that roughly predicts the number of cells over time. This type of modeling has no microenvironment, no immune system, no circulatory system, nothing except cells undergoing proliferation or death and possibly mutation. The rate of proliferation and the rate of death are numbers determined by a dose-response equation for that treatment. The dose-response equation is taken from textbook toxicology for now, but that part is easy to customize. You can swap in your favorite. Anyway, a tumor is just a number of cells, and if the number shrinks to 0, then the disease is cured, the end. If the number grows larger than 10 to the 12, then that's considered a lethal tumor burden, and it's also the end of the simulation. One last assumption is that a cell can change its level of drug sensitivity just by mutation. After this change, the cell shifts left or right along the dose-response curve, which gives it a new proliferation rate and death rate. These rates are inherited by any daughter cells that occur, and that's it. Those are the assumptions, and if it seems too boring, just consider combination therapy. If there are two drugs, then we have two dimensions of dosing on a dose-response landscape, and the two drugs can be additive or non-additive. We call drugs synergistic if their efficacy is greater than additive. Our first question about combination therapy is the most simple. Are synergistic combination drugs better than additive at prolonging survival time during long-term evolution? We developed a general mathematical answer to the question, which you shouldn't try to read in a slideshow, but my point is that it is possible to delineate a general form of the solution. The essence of what we found in that work is that synergistic combination drugs have a pitfall, which we call the paradox of synergism. Although synergistic combinations are very effective at achieving strong control toward the initial population of sensitive cells, the blue cells, the problem is they lose efficacy more steeply than an additive combination as soon as some drug resistance arises. In the worst case, synergistic combinations can accelerate the evolution of double drug resistance. To understand why that is, the definition of synergism says there's an extra strong effect when both drugs are used together. That means a cell would have to fully experience both drugs to have the strong effect, and if the cell becomes insensitive to one of the drugs, it escapes the mountaintop and gets a huge reward in evolutionary fitness. The number of cells over time would be affected by that reward. To put it quantitatively, plotting number of cells over time, the vast majority of cells are blue cells getting killed by the treatment, and synergism makes that stronger. But not all cells are blue. There's a small number of red cells that are resisting drug A. They can persist or maybe grow a bit during the combination treatment, and that's where synergism gives them the larger reward. If these cells aren't growing very fast, then why is it so bad to let them live? The math says it is important to kill as many red cells as possible, because as soon as you have even one doubly resistant cell, the black type, that would be a death sentence. And each red cell is just one mutation away. And so it's crucial to minimize the number of red cells so that we minimize the number of opportunities for that last transition to occur. What happens to the blue cells is less important unless the blue cells can be decimated as a way of preventing the creation of red cells. In summary, synergistic combinations have a weakness against long-term evolution of resistance. That's number one, the paradox of synergism. That's already been published, so I want to spend the rest of my time presenting unpublished work. Number two, what if drug resistance has levels like percent resistance? That turns out to give very different long-term evolution compared with all or nothing resistance. And number 3, what if we have more than 2 drugs? To create levels of drug resistance, we take the transition from blue to red and lay it out as multiple gradations shown in purple. If we do that for both drug A and drug B, we have a 2-dimensional grid of partial resistance. Each arrow is a transition, which could be a mutation or an epigenetic or regulatory change. Each transition occurs with some small probability called the transition rate. As before, the proliferation rate and death rate of each state is extrapolated from the dose-response curve. And if we feed this into a simulation of evolution, what we see is, at the top in purple, if you need multiple hits to achieve single drug resistance, like multiple mutations, that creates more obstacles to full resistance. And as we increase the number of obstacles, we get larger differences in outcome between additive or synergistic combination therapies. Whereas to the left in yellow, if the landscape of resistance is highly plastic, such as having tiny gradual changes that don't require an infrequent mutational event, then it stops mattering whether the treatment is an additive combination or a synergistic combination. Of course, the efficacy still matters, but it no longer matters how that efficacy is achieved through synergism or additivity. Next, we studied what paths cells take for achieving resistance as they transition from a fully sensitive state on the left toward more resistant states toward the right. If cells can change along various paths, which paths will give them the greatest proliferation or the least death? Do cells tend to go through the middle of the grid, through states that are equally resistant to both drugs? The green states are called symmetric resistance. Or do they tend to develop one-sided resistance toward one drug more than the other, like the blue states called asymmetric resistance? We did many simulations across a spectrum of synergistic and non-synergistic combination therapies and counted how many cells were on each type of escape path as a ratio of green to blue. That's the y-axis, which we plotted over time for synergistic versus non-synergistic combination therapies. This shows that as we choose more and more synergistic combinations as the alpha parameter gets larger, the escape path gets more asymmetric, more one-sided against one drug than the other. So that means when resistance arises after treatment with a combination therapy, if the combination was synergistic, then the most likely resistance will be against one drug more than the other. Whereas if the combination treatment was additive or non-synergistic, then the escape path was more likely to be green, where both drugs are compromised. So now I've shown how gradations and granularity of drug resistance affect the evolutionary outcomes. Next, let's look at combinations of 3 or more drugs. Here are 8 categories of phenotype ranging from fully sensitive on the left to fully resistant on the right, with all permutations of single and double resistance in between. Let's simulate evolution using a fair comparison of different combination strategies. This is as fair as we can get. We're choosing between a cocktail of drugs A plus B plus C versus a cocktail of D plus E plus F, and each individual drug has the exact same efficacy, and each 3-drug cocktail has the exact same efficacy. We ask, does it help or hurt the long-term evolution to have different pairwise effects within the 3-drug cocktail? For example, cocktail ABC has equally effective pairs, all of them additive, whereas cocktail DEF has one synergistic pair, one antagonistic pair, one additive pair. The answer is that everything matters. The uniformity of the pairwise drug interactions and the efficacy of the 2-drug subsets do affect the evolution of drug resistance. These plots show cell numbers over time. The y-axes are log-scaled, so the exponential growth looks linear. At first, the total number of cells in yellow shrinks due to treatment, but then in pink, some partially resistant subclones arise and expand, and when there's enough of them, one of them mutates into a multi-resistant phenotype in navy blue, which has a really fast slope of clonal expansion, and that creates a lethal tumor burden. The multi-drug resistance is only dominant at the end, but its emergence is much earlier, and the reservoir of partial resistance that allows it to emerge is even earlier. If the 3-drug cocktail has unequal efficacy for the 2-drug subsets, then the weakest subset creates the favorite path for escape. Hands down, that's despite having equal cocktail efficacy and equal single drug efficacy. What that means is we need to design our 3-drug cocktails so that every 2-drug subset gives effective control. And if we build 3-drug combinations by augmenting our favorite 2-drug combination, we might feel happy knowing the cocktail contains a really awesome 2-drug subset. But that won't help the patient because the fantastic subset of drugs is not the escape path that most needs to be blocked. The most popular escape path is always the unguarded exit. We've seen what allows cocktails to fail, so now let's work that knowledge for leverage. Suppose we know there is a weak link. Suppose we know that one of our drugs is easy to resist. For example, we have a BRAF mutant cancer we're treating with vemurafenib, and we believe resistance will develop against vemurafenib more quickly than against the other drugs. We can simulate that by letting one of the transitions, resistance to drug A, have faster probability, simulate that, and the plot, if we look at the time span until developing a lethal tumor burden on the y-axis and the mutation rate on the x-axis, if the mutation rate for vemurafenib resistance is really slow, then the best strategy is just uniform synergism or uniform additivity, uniform across the whole cocktail. But if the mutation rate gets higher, in this case 10 to the negative 4, then the winning strategy is to strengthen drug B and C synergism, make them more highly synergistic with each other, while drugs A plus B and A plus C compensate with the opposite. We've now demonstrated a technology that can change how we think about the job of preventing, delaying, and managing the evolution of drug resistance. In summary, I've explained how the paradox of synergism arises for pairs of drugs, and then looking at graded resistance, I showed how the spacing and levels of resistance can create or remove bottlenecks in the phenotypic landscape of drug resistance. More bottlenecks makes synergism more impactful, and fewer bottlenecks makes it less important. We also traced the likely escape paths for cancer cells to develop resistance and found that synergistic combination drugs are more likely to be escaped using asymmetric resistance, where one drug is more compromised than the other. Finally, I talked about 3-drug cocktails, where we found that escape paths develop wherever defenses are weakest. The strength of the strongest subsets hardly affects the duration of disease control at all, and the most important consideration is the weakness of the weakest subsets of the drugs. There are a lot of caveats for interpreting this type of modeling. The main thing is that it's purely theoretical. It's based on simplified models of reality that don't consider the immune system, the microenvironment, or much of what we know and love about in vivo biology. Another simplification is that all traits are heritable and independent of each other. For future work, we want to apply these insights to specific systems. People in the audience may be able to suggest good test beds. We've built a platform that can simulate scenarios, so now it's time to do those simulations. If you have non-obvious trade-offs that you want us to predict, questions to answer, please get in touch. I'm the only person on Google named Lisa Tucker-Kellogg. Finally, it's with great appreciation that I acknowledge the talented students I've been privileged to work with on this project, especially Alicia Saputra and Vivek Murali. I also thank the funding agencies that have supported this work, and thank you for your time and attention.